Diseases of banana. First is yellow sigatoka leaf spot by Mycosferella musicola. This is the sexual stage, and Pseudosarcospora musi. This is the asexual stage. Became epidemic in 1913 in Sigatoka Valley, Fiji. Early symptoms on leaves: small spikes, later increase in size, turned into spindle-shaped spikes on leaves near tip and near margin of the lamina. Spot squaless, drying of entire spotted area. Other symptoms: thin brownish streaks darken and expand into large areas of necrotic tissue. Thin brownish streaks darken and expand into large areas of necrotic tissue. Mature lesions produce sexual structures. So yellow sigatoka disease is caused by Pseudosarcospora musi, that is the asexual stage, and the sexual stage is Mycosferella musicola. Pseudosarcospora musi, asexual stage, Mycosferella musicola. Then etiology: ascospores and conidia. Pseudothecium. Ascospores: surface moisture release. 3 types of etiology. Ascospores: dispersed by wind over long distances to new plants, infect plant debris. Pseudothecium, and then conidia. Conidia windborne and spread the disease. So 3 types of etiology are produced by yellow sigatoka leaf spot causing fungi in banana. First is ascospores, second is pseudothecium, and third is conidia. Ascospores, pseudothecium, conidia. Spermatia produce spermagonium or spermagonia into neighboring. Fertilization between neighboring spermagonium into neighboring receptive hyphae produce pseudothecia or pseudothecium that carry ascospores. Ascospores infect the leaves through stomata. Conidiophores emerge through stomata. Conidiophores produce conidia, infect the leaves through stomata. So spermatia produce spermagonium. Spermagonium or spermagonia carry spermatia. Fertilization between spermagonium into neighboring receptive hyphae produce pseudothecia or pseudothecium that carry ascospores, infect the leaves through stomata. Emerge through stomata. Conidiophores produce conidia that infect the leaves through stomata. Management: cultural: planting of banana in well-drained soil, removal and destruction of affected leaves, grow resistant varieties like Carpura chakkerakeli. Chemical spray: Botox mixture @1% plus linseed oil @2% or mancozeb or chlorothalonil @0.1%. Resistant varieties. What is the resistant varieties against yellow sigatoka leaf spot of banana? That is the Carpura chakkerakeli. Carpura chakkerakeli variety is the resistant variety against yellow sigatoka leaf spot of banana. Then Panama wilt. Panama wilt is caused by Fusarium oxysporum forma specialis cubans. It was first reported in Australia in 1874. Panama wilt is caused on 3 to 5 months old plant. Yellow streaks, longitudinal splitting of the pseudostem. This is the characteristic symptom, typical characteristic symptom. Longitudinal splitting of pseudostem. Rhizomes have rotten fish odor. So Panama wilt of banana is caused by Fusarium oxysporum forma specialis cubans. Fusarium oxysporum forma specialis cubans was first reported in Australia in 1874 on 3 to 4 months old plant. Yellow streaks, longitudinal splitting of pseudostem, typical characteristic symptom. Rhizomes have rotten fish odor. Primary source of inoculum or primary source of infection: chlamydospores in soil, propagules in infected suckers used for planting. And secondary source of infection: macro and microconidia through irrigation water. Epidemiology: infection by burrowing nematode Rhodophila similis predispose the disease and poorly drained soil. So 2 types of etiology are there. First is chlamydospore, second is macro and microconidia. Primary source of inoculum: chlamydospores in soil or propagules in infected suckers used for planting, and secondary source of inoculum is the macro and microconidia through irrigation water. Epidemiology: infection by burrowing nematode that predispose the disease in the soil, that is Rhodophila similis, and poorly drained soil. Resting spores formation in the drying plants. Resting spores germination in the soil. Infection from root to root contact, movement up xylem. Resting spores formation in the drying plant. Resting spores germination on the soil. Infection from root to root contact, movement up to xylem. Flood fallowing for 6 to 24 months. Apply lime at the rate 1 to 2 kg per peat. Dipping of suckers in carbendazim at the rate 0.1% solution before planting. Apply neem cake plus trichoderma viridi in peats. Resistant varieties, Cavendish varieties. Flood fallowing for 6 to 24 months. Resistant varieties, Cavendish varieties of banana. Third type of disease of banana is Moco disease, that is caused by Ralstonia solanacearum. That is the bacteria. First reported in Guyana, not reported in India. This is aerobic, gram-negative, non-spore forming, soil-borne, motile with a polar flagellum tuft. Moco disease is caused on 3 to 4 leaves of young plants. Discoloration of vascular bundles. This is the characteristic symptom. Bacterial ooze, greyish-brown bacterial ooze on the pseudostem, cut transversely, colonize the xylem vessels. Epidemiology, slightly acidic soil and nematode infection. So Moco disease of banana is caused by Ralstonia solanacearum. Ralstonia solanacearum is the bacteria that is gram-negative aerobic, non-spore forming, motile, soil-borne with a polar flagellum tuft. Was first reported in Guyana, not reported in India. Which type of disease of banana is not reported in India? That is Moco disease of banana. Moco disease of banana is found on 3 to 4 leaves of young plants. Discoloration of vascular bundle, it is the characteristic typical symptom. Bacterial ooze is the greyish brown in color on the pseudostem that cut transversely. This type of bacteria colonizes the xylem vessels. Epidemiology, slightly acidic soil and nematode infection that predispose the disease. Crop rotation with sugarcane or rice. Soil drenching with 1.5% of bleaching powder solution or 1% of Bodox mixture or 0.02% of streptocycline. Which type of antibiotic is used against Moco disease of banana? Streptocycline antibiotic. Crop rotation with which type of crops? Against Moco disease of banana, sugarcane and rice. Airvinia rhizome rot or soft rot. Airvinia rhizome rot or soft rot is caused by Airvinia ceratovora or Airvinia chrysanthemi, that is the bacteria. Was first reported in Honduras by Gross Michael in 1949. In India, disease incidence 70% in Karnataka. Airvinia rhizome rot. Airvinia rhizome rot or soft rot is caused by Airvinia ceratovora or Airvinia chrysanthemi, that is the bacteria. Was first reported in Honduras by Gross Michael in 1949. In India, disease incidence 70% in Karnataka. Airvinia rhizome rot or soft rot by Airvinia ceratovora, Airvinia chrysanthemi bacteria. Airvinia rhizome rot or soft rot is cause damage on young plants, toppling of germinating suckers, infection on pseudostem, rhizome, scanty roots. This bacteria is Peritricus gram-negative. Epidemiology is a susceptible juvenile stage of the crop, waterlogging soil, high 37 degrees Celsius temperature in early crop growth stages. So what are the infections? Infections on young plants, toppling of germinating suckers, infection on pseudostem, rhizome and scanty roots. These bacteria, there's Airvinia ceratovora and Airvinia chrysanthemi, gram-negative Peritricus bacteria. Epidemiology, juvenile crop stage is the most susceptible stage of this disease. Waterlogging condition of the soil and high 37 degrees Celsius temperature in early crop growth stages. Primary source of infection by bacterial cell in soil and diseased suckers. Secondary source of infection by bacterial cells through irrigation water. Primary source of infection by bacterial cell in soil and diseased suckers. Secondary infection by bacterial cells through irrigation water. Drenching of plant base with Pseudomonas fluorescens. Drenching at the rate 1 to 2 liter per plant. Dip the suckers in copper oxychloride at the rate 2.5 milliliter per liter of solution. Crop rotation with rice or sugarcane for up to 3 to 4 years. So crop rotation with which type of crops? Rice or sugarcane for 3 to 4 years. Fifth disease of banana is Bunchita or curly top or cabbage top. This is caused by BBTV or banana Bunchita virus, has single-stranded DNA. Was first reported in Fiji in Cavendish varieties. Which type of varieties was first? Bunchy top or curly top or cabbage top or disease of banana was first reported in Cavendish varieties in Fiji. Introduced in India from Sri Lanka by cyclones. Infection at any stage, intervenal chlorosis, pale lamina, Morse code. Morse code is the typical characteristic symptom of curly top or bunchy top or cabbage top, bunchy top formation. This type of virus is a domestic quarantine pest in India. So bunchy top or curly top or cabbage top disease of banana is caused by BBTV or banana bunchy top virus. Was first reported in Fiji in Cavendish varieties. This virus has single-stranded DNA. Introduced in India from Sri Lanka through cyclones. Infection at any stage, intervenal chlorosis, pale lamina. Morse code is the main characteristic typical symptom of bunchy top or curly top or cabbage top disease of banana. And bunchy top formation. This banana bunchy top virus is a domestic quarantine pest in India. Has single-stranded DNA. Primary source of inoculum is virus particles by diseased suckers, and secondary source of inoculum, this virus is transmitted by banana black aphid. Banana black aphid, that is Pentalonia nigronervosa. Use only certified suckers. This aphid is controlled by methyl dimeton at the rate 2 ml per liter or phosphomethone at the rate 1 ml per liter. So banana bunchy top virus is transmitted by banana black aphid, that is Pentalonia nigronervosa. So primary source of inoculum by virus particles by diseased suckers, and secondary source of inoculum by aphids, that is the vector. Last disease or sixth disease of banana is banana mosaic or infectious sclerosis or heart rot. Banana mosaic or infectious sclerosis or heart rot by CMV or cucumber mosaic virus. It is a domestic quarantine pest in India, means movement of planting suckers from Gujarat and Maharashtra is banned to other parts of the country or India. Transmitted by aphids, two species of aphids, Apis maydis and Apis gossypi. Mosaic, banana mosaic or infectious sclerosis or heart rot is caused by CMV or cucumber mosaic virus. That is a quarantine pest in India, domestic quarantine pest in India. Movement of planting suckers from Gujarat and Maharashtra is banned to other parts of India or the country. This is transmitted by aphids, two species of aphids, Apis maydis and Apis gossypi. So what are the other names of the banana mosaic disease? Other names are banana mosaic disease are infectious sclerosis and heart rot, caused by CMV or cucumber mosaic virus. The symptoms are mosaic, discontinuous streaking bands from leaf margin to midrib, parallel to veins. Heart rot, rotting of central youngest leaves and leaf sheath. Primary source of inoculum, diseased suckers, and secondary source of inoculum, vector aphids. So what are the symptoms? Damage symptoms: mosaic, discontinuous streaking bands from leaf margin to midrib, parallel to veins. Heart rot, rotting of central youngest leaves and leaf sheath. Primary source of inoculum, diseased suckers, and secondary source of inoculum, vector aphids. So controlled by dry heat treatment at 40 degrees Celsius for 1 day, and vector aphids is controlled by methyl dimeton at the rate 2 ml per liter. Dry heat treatment. Which type of treatment? Dry heat treatment at 40 degrees Celsius for 1 day, and aphid or vector is controlled by methyl dimeton at the rate 2 ml per liter. 40 degrees Celsius for 1 day.